Good morning everyone. In this video we are going to discuss about a topic from the course Biomedical Instrumentation. The title is Biomedical Electrodes. So first of all, let's know what is an electrode. So as we know, an electrode is a device that converts the biomedical signal into an electronic signal. So for measuring the bioelectric signals from the human body, we are using various types of electrodes in the medical field. So what are the various types of electrodes? We are going to discuss in detail here. So these are the various types of electrodes used in biomedical instrumentation. The first one is surface electrode, then microelectrodes. The microelectrodes, again it is converted into two types, that is metal microelectrode and non-metallic microelectrode. So it is also called as micropipette. And the third one is needle electrodes. So let's discuss in detail about all these three types. First one is surface electrode. So as the name indicates, we are placing the electrode over the surface of the skin to measure the bioelectric signals. So we all know about ECG, EEG, EMG measurements, where we are measuring the bioelectric signal from the heart, brain and the muscles of human body. So for all these measurements, we are using this surface electrodes. So we are having many types of surface electrodes, and it is listed here: the metal plate electrode, suction cup electrode, adhesive type electrode, floating electrode, ear clips and scalp electrode. Let's see one by one. So this is the image of a metal plate electrode. So here we are having a metal plate which is made up of any metal. This plate will be placed over the surface of the skin. So how we are placing it in the skin is by using a electrolyte gel or a electrolyte paste. So they will be placing the paste over the place where we have to place the electrode, and over the paste they will be placing the metal electrode. So here we are having a disadvantage. Since we are using this paste, electrolyte paste, there may be a movement or dislocation of this metal plate electrode. Because of that, there will be the occurrence of measurement errors. So that is the main disadvantage of metal plate electrodes. So next one is the suction cup electrode. So this is the physical appearance of a suction cup electrode. So as you see in the image, we are having a contact surface, and here we are having a squeezable rubber bulb, and here we are having the lead wire by which we are going to measure the bioelectric signal. So this is mainly used in ECG measurement. Okay, the main point is this suction cup electrodes we can use only in the flat surface of the body or the body parts where soft tissues are available. So only in that particular surface we can prefer the suction cup electrodes. So only in ECG measurement we are using this suction cup electrode. But in practical, the infection possibility is high, and also the cleaning process is very tedious. So nowadays they are not preferring suction cup electrodes for measuring the bioelectric signals. Then third one is adhesive type electrode. So first one we discussed about the metal plate electrode. I told you that we are using a electrolyte paste or an electrolyte gel to fix the electrode on the surface or the skin surface. So in this adhesive type electrode, already a pad like structure will be there in the bottom side of the electrode. So in there a preloaded adhesive or a gel will be available which can be placed over the skin. So there won't be any movement of the electrode during the measurement, so we can avoid the measurement error. So it is an advantage when we compare to metal plate electrode. Then next one is the floating electrode. So here we are using a electrolyte jelly like substance to place the electrode over the skin surface. So here also there won't be any movement of the electrode, which avoids the measurement errors which occurs in metal plate electrode. Then last one is ear clips and scalp electrode. So these types of electrodes are mainly used in EEG measurement, in which we are measuring the bioelectric signal from the brain. So around the ear region they are using ear clips, and over the skull area they are using the scalp electrodes during the EEG measurement. So these are the 5 different types of surface electrodes used for bioelectric signal measurement. Then next we are moving to the micro electrodes. So as the name indicates, micro electrodes. So we can measure the bioelectric signal from a single cell. So for that purpose we are using micro electrodes. So micro electrodes we are having two types: metal microelectrode and non-metallic microelectrode. So metal microelectrode, the tip of the electrode will be made up of any metallic substance. So that's why we are calling it as the metal microelectrode. So this is the image of a metallic microelectrode. So the diameter, the tip of the microelectrode will be very small, so that it can penetrate easily inside the skin. So here microelectrodes, we are using two electrodes. This is the microelectrode and this is the reference electrode. So both the electrodes will be measuring the potential from the human body. The difference between the potential of both the electrodes will give the resultant output during the use of microelectrode. This is the non-metallic microelectrode, or it is called as the micropipette. Here the tip of the microelectrode will be made up of non-metallic substance. That why we are given the name as non-metallic microelectrode. So a glass pipette will be there, in which the electrolyte solution will be filled. Here also we are using two different electrodes. One is the microelectrode and another one is the reference electrode. So both the electrodes will be measuring the bioelectric potential. Then finally the difference between both the potential will give us the resultant output signal. Then last type of electrode is the needle electrode. It is similar to the syringe which is used in the medical field. So this is mainly used for the biosignal measurement from a nerve, from a single nerve or a particular portion of a nerve. We can measure the bioelectric signal by using the needle electrode. So this is the portion which we are injecting inside. The other end we are having a bent wire. So once we are injecting this needle inside the body, this lead wire or the bent wire will be placed in the particular portion where we have to measure the signal. So after inserting, the lead wire will be placed in the particular part and the needle will be taken outside. So after that we will be measuring the bioelectric signal by using this lead wire, which is placed inside the body. So this is how we are using the needle electrodes. So needle electrode also we are having two different types, that is monopolar electrode and bipolar electrode. So these are about the different types of electrodes which we are using in the biomedical field for measurement of the bioelectric signal from various parts of our body. Various parts in the sense uh, brain, heart, muscles, nerves, and even from a single cell of a human body, we can measure the bioelectric signal by using these different types of microelectrodes. Thank you.